Hello students, today we'll be studying the tricarboxylic acid or the TCA cycle or the Krebs cycle. Let's begin with the introduction. The metabolism of glucose to pyruvate in glycolysis, which is an anaerobic process, harvests just a fraction of the ATP available from glucose. Most of the ATP generated in metabolism is provided by the aerobic processing of glucose. This process starts with the complete oxidation of glucose derivatives to carbon dioxide. This oxidation takes place in a series of reactions called the citric acid cycle, also known as the tricarboxylic acid or TCA cycle or the Krebs cycle. The citric acid cycle is a final common pathway for the oxidation of fuel molecules, carbohydrates, fatty acids, and amino acids. Let's look at the overview of the citric acid cycle. The citric acid cycle is the central metabolic hub of the cell. It is the gateway to the aerobic metabolism of any molecule that can be transformed into an acetyl group or a component of the citric acid cycle. The cycle is also an important source of precursors for the building blocks of many other molecules such as amino acids, nucleotide bases, and porphyrin, which is the organic component of heme. The citric acid cycle component, oxaloacetate, is also an important precursor to glucose. So what is the function of the citric acid cycle in transforming fuel molecules into ATP? Fuel molecules are carbon compounds that are capable of being oxidized by losing electrons. The citric acid cycle includes a series of oxidation-reduction reactions that result in the oxidation of an acetyl group to two molecules of carbon dioxide. This oxidation generates high-energy electrons that will be used to power the synthesis of ATP. The function of the citric acid cycle is the harvesting of high-energy electrons from carbon fuels. The overall pattern of the citric acid cycle is shown here. A 4-carbon compound, oxaloacetate, condenses with a 2-carbon acetyl unit to yield a 6-carbon tricarboxylic acid. The 6-carbon compound releases carbon dioxide twice in 2 successive oxidative decarboxylations that yield high-energy electrons. A 4-carbon compound remains. This 4-carbon compound is further processed to regenerate oxaloacetate, which can initiate another round of the cycle. 2 carbon atoms enter the cycle as an acetyl unit and 2 carbon atoms leave the cycle in the form of 2 molecules of carbon dioxide. The citric acid cycle itself neither generates a large amount of ATP nor includes oxygen as a reactant. Instead, the citric acid cycle removes electrons from acetyl-CoA and uses these electrons to form NADH and FADH2. 3 hydride ions, hence 6 electrons, are transferred to 3 molecules of NAD+, and 1 pair of hydrogen atoms, hence 2 electrons, is transferred to 1 molecule of flavin adenine dinucleotide or FAD. These electron carriers yield 9 molecules of ATP when they are oxidized by molecular oxygen in oxidative phosphorylation. Electrons are released in the reoxidation of NADH and FADH2 flow through a series of membrane proteins, which are referred to as the electron transport chain, to generate a proton gradient across the membrane. These protons then flow through ATP synthase to generate ATP from ATP and inorganic phosphate. The citric acid cycle, in conjunction with oxidative phosphorylation, provides the vast preponderance of energy used by aerobic cells in human beings, greater than 90%. It is highly efficient because the oxidation of a limited number of citric acid cycle molecules can generate large amounts of NADH and FADH too. Before we go into the reactions of the citric acid cycle, let us study the reactions that links glycolysis to the citric acid cycle. The enzyme pyruvate dehydrogenase links glycolysis to the citric acid cycle. Carbohydrates, mainly glucose, are processed by glycolysis into pyruvate. Under anaerobic conditions, the pyruvate is converted into lactate or ethanol depending on the organism. Under aerobic conditions, the pyruvate is transported into the mitochondria by a specific carrier protein embedded in the mitochondrial membrane. In the mitochondrial matrix, pyruvate is oxidatively decarboxylated by pyruvate dehydrogenase complex to form acetyl-CoA. This irreversible reaction is a link between glycolysis and the citric acid cycle. The pyruvate dehydrogenase complex produces carbon dioxide and captures high transfer potential electrons in the form of NADH. Thus, the pyruvate dehydrogenase reaction has many of the key features of the reactions of the citric acid cycle itself. Now let's study the reactions of the citric acid cycle. Citrate synthase forms citrate from oxaloacetate and acetyl coenzyme A. The initial reaction between acetyl-CoA and oxaloacetate to form citrate is catalyzed by citrate synthase, which forms a carbon-carbon bond between the methyl carbon of acetyl-CoA and the carbonyl carbon of oxaloacetate. The thioester bond of the resultant citral-CoA is hydrolyzed, releasing citrate and CoAsh, an exergonic reaction. Citrate is isomerized into isocitrate. Citrate is isomerized to isocitrate by the enzyme aconitase or aconitase hydratase. The reaction occurs in two steps: dehydration to cis-aconitate, some of which remains bound to the enzyme, and rehydration to isocitrate. The hydroxyl group is not properly located in the citrate molecule for the oxidative decarboxylations that follow. Thus, citrate is isomerized into isocitrate to enable the 6-carbon unit to undergo oxidative decarboxylation. The isomerization of citrate is accomplished by a dehydration step followed by a hydration step. The result is an interchange of an hydrogen and an hydroxyl group. The enzyme catalyzing both steps is called aconitase because cis-aconitate is an intermediate. Next step is isocitrate is oxidized and decarboxylated to alpha-ketoglutarate. We come now to the first of 4 oxidation reduction reactions in the citric acid cycle. The oxidative decarboxylation of isocitrate is catalyzed by isocitrate dehydrogenase. Isocitrate undergoes dehydrogenation to form initially oxalosuccinate, which remains enzyme bound and undergoes decarboxylation to alpha-ketoglutarate. The decarboxylation requires magnesium or manganese ions. This oxidation generates the first high transfer potential electron carrier, NADH, in the cycle. Next step is succinyl coenzyme A is formed by the oxidative decarboxylation of alpha-ketoglutarate. The conversion of isocitrate into alpha-ketoglutarate is followed by a second oxidative decarboxylation reaction, the formation of succinyl-CoA from alpha-ketoglutarate. This reaction is catalyzed by a multi-enzyme complex, the alpha-ketoglutarate dehydrogenase complex. This enzyme complex is similar to that involved in the oxidative decarboxylation of pyruvate. That is, it requires the same cofactors as a pyruvate dehydrogenase complex, which is thiamine diphosphate, lipoate, NAD+, FAD, and coenzyme A, and results in the formation of succinyl-CoA. This is the only example in the citric acid cycle of substrate-level phosphorylation. A compound with high phosphoryl transfer potential, GDP or ADP, is generated from succinyl coenzyme A. Succinyl-CoA is an energy-rich thioester compound. In the citrate synthase reaction, the cleavage of the thioester bond powers the synthesis of the 6-carbon citrate from the 4-carbon oxaloacetate and the 2-carbon fragment. But here, the cleavage of the thioester bond of succinyl-CoA to yield succinate and CoA is coupled to the phosphorylation of a purine nucleoside diphosphate, usually GDP. This reaction is catalyzed by succinyl-CoA synthetase or succinate thiokinase. This reaction is the only step in the citric acid cycle that directly yields a compound with high phosphoryl transfer potential. Next, oxaloacetate is regenerated by the oxidation of succinate. Reactions of 4-carbon compounds constitute the final stage of the citric acid cycle, the regeneration of oxaloacetate. The onward metabolism of succinate leading to the regeneration of oxaloacetate is the same sequence of chemical reactions as occurs in the beta oxidation of fatty acids. That is dehydrogenation to form a carbon-carbon double bond, addition of water to form a hydroxyl group, and a further dehydrogenation to yield the oxo group of oxaloacetate. The first dehydrogenation reaction, forming fumarate, is catalyzed by succinate dehydrogenase, which is bound to the inner surface of the inner mitochondrial membrane. The enzyme contains FAD, an iron sulfur protein, and directly reduces ubiquinone in the respiratory chain. Fumarase, also known as fumarate hydratase, catalyzes the addition of water across the double bond of fumarate, yielding malate. Malate is converted to oxaloacetate by malate dehydrogenase, a reaction requiring NAD+. Although the equilibrium of this reaction strongly favors malate, the net flux is towards the direction of oxaloacetate because of the continual removal of oxaloacetate, either to form citrate as a substrate for gluconeogenesis or to undergo transamination to aspartate, and also because of the continual reoxidation of NADH. Now let's see how 12 ATP molecules are formed per turn of the citric acid cycle. As a result of oxidations catalyzed by the dehydrogenases of the citric acid cycle, 3 molecules of NADH and 1 of FADH2 are produced for each molecule of acetyl-CoA catabolized in 1 turn of the cycle. These reducing equivalents are transferred to the respiratory chain, where reoxidation of each NADH results in formation of 3 ATP and the reoxidation of FADH2 in formation of 2 ATP. In addition, 1 ATP or GTP is formed by substrate level phosphorylation, which is catalyzed by succinate thiokinase. So the net reaction of the citric acid cycle is acetyl-CoA plus 3 NAD plus FAD plus GDP plus inorganic phosphate plus 2 molecules of water gives 2 molecules of carbon dioxide plus 3 NADH plus FADH2 plus 1 GTP plus 2 protons and 1 CoA. Vitamins play key roles in the citric acid cycle. Let's see how. 4 of the B vitamins are essential in the citric acid cycle and therefore in energy-yielding metabolism. Number 1, riboflavin in the form of flavin adenine dinucleotide, FAD, a cofactor in the alpha-ketoglutarate dehydrogenase complex and in succinate dehydrogenase. Number 2, niacin in the form of nicotinamide adenine dinucleotide or NAD, the coenzyme for 3 dehydrogenases in the cycle, isocitrate dehydrogenase, alpha-ketoglutarate dehydrogenase, and malate dehydrogenase. Number 3, thiamine, which is vitamin B1, as thiamine diphosphate, the coenzyme for decarboxylation in the alpha-ketoglutarate dehydrogenase reaction. And number 4, pantothenic acid, as part of the coenzyme A, the cofactor attached to active carboxylic acid residues such as acetyl-CoA and succinyl-CoA. The citric acid cycle plays a pivotal role in metabolism. The citric acid cycle is not only a pathway for oxidation of 2 carbon units, it is also a major pathway for interconversion of metabolites arising from transamination and deamination of amino acids. It also provides the substrates for amino acid synthesis by transamination as well as for gluconeogenesis and fatty acid synthesis. Because it functions in both oxidative and synthetic processes, it is amphibolic. Now let's look at the regulation of the citric acid cycle. In most tissues where the primary role of the citric acid cycle is in energy-yielding metabolism, respiratory control via the respiratory chain and oxidative phosphorylation regulates the citric acid cycle activity. Thus, activity is immediately dependent on the supply of NAD+, which in turn, because of the tight coupling between oxidation and phosphorylation, is dependent on the availability of ATP and hence ultimately on the rate of utilization of ATP in chemical and physical work. In addition, individual enzymes of the cycle are regulated. The most likely sites for regulation are the non-equilibrium reactions catalyzed by pyruvate dehydrogenase, citrate synthase, isocitrate dehydrogenase, and alpha-ketoglutarate dehydrogenase. The dehydrogenases are activated by calcium ions, which increases in concentration during muscular contraction and secretion, when there is increased energy demand. In a tissue such as brain, which is largely dependent on carbohydrate to supply acetyl-CoA, control of the citric acid cycle may occur at pyruvate dehydrogenase. Several enzymes are responsive to the energy status, as shown by the ATP-ADP ratio and the NADH-NAD+ ratios. Thus, there is allosteric inhibition of citrate synthase by ATP and long-chain fatty acyl-CoA. Allosteric activation of mitochondrial NAD-dependent isocitrate dehydrogenase by ATP is counteracted by ATP and NADH. The alpha-ketoglutarate dehydrogenase complex is regulated in the same way as is pyruvate dehydrogenase. Succinate dehydrogenase is inhibited by oxaloacetate, and the availability of oxaloacetate as controlled by malate dehydrogenase depends on the NADH and NAD+ ratio. Since the Km for oxaloacetate of citrate synthase is of the same order of magnitude as intramitochondrial concentration, it is likely that the concentration of the oxaloacetate controls the rate of citrate formation. Finally, let's come to the conclusion. The citric acid cycle is a final pathway for the oxidation of carbohydrates, lipids, and proteins whose common end metabolite acetyl-CoA reacts with oxaloacetate to form citrate. By a series of dehydrogenations and decarboxylations, citrate is degraded, releasing reduced coenzymes and molecular carbon dioxide and regenerating oxaloacetate. The reduced coenzymes are oxidized by the respiratory chain linked to the formation of ATP. Thus, the cycle is the major route for the generation of ATP and is located in the matrix of mitochondria, adjacent to the enzymes of the respiratory chain and oxidative phosphorylation. The citric acid cycle is amphibolic, since in addition to oxidation, it is important in the provision of carbon skeletons for gluconeogenesis, fatty acid synthesis, and interconversion of amino acids, and thus play a pivotal role in metabolism.